starting with what? We are starting with animal viruses. So we learned about the viruses that infect to what? That infect to the bacteria. We call them what? Bacteriophages. We call them bacteriophages. Now let's learn the animal viruses. So these viruses, animal viruses, these are viruses that infect to what? That infect to the animal cells in your body. Think of them as a tiny invaders trying to make copies of themselves inside of you. Okay? So normally some viruses burst cell open. So this is called the lysis. They're going to lyse the cell and they're going to go outside to escape. Do you remember this process, lysis? For example, in bacteria, if the virus is going to enter, if the virus is inside of the bacteria, they're going to burst the cell. They're going to burst the bacterial cell, lyse the bacterial cell. This this kill the cell. Just it kills the cell, just like popping a balloon. So, but animal viruses, they have a smarter way. Okay, they butt out of the cell without killing it. Do you see butting of the virus from the cell? This is the animal cell. As you can see here, this is the animal cell, and this is the virus which is butting out from the cell. This butting out of the cell without killing it. So, but the animal cell is staying alive. It's not killing the animal cell here. Okay? So viruses that infect the animal cells can simply bud out through the plasma membrane instead of lighting the cell to escape. Okay? So what is budding trick actually? Imagine the virus is a spy sneaking out the house. I mean the cell here. Okay? Instead of breaking the windows when they are running out, when the spy is going to run out, instead of breaking down the window, it just slips out the front door quietly and gently. Okay, so how is this happening? How this process is happening? First of all, the virus. This is the virus. Okay, this virus attached to the cell using their ligand. Okay, this is the ligand. Using these ligands, there are a lot of ligands, they're going to attach to the receptors. You see the red ones are receptors and the bluish colorations are the ligands. So ligands, they are the keys, okay? They are the keys. And the receptors, they are what? They are locks, okay? So receptor is like a lock and the ligand is like a key. So the virus enters to the cell then, okay? Then the virus is entering when the key and the lock, when they going to match together, this virus enters the cell and uses the cell's own machinery to make more viruses inside of the cell, okay? Like making photocopies a lot. So using these cells machinery, what's the meaning of cells machinery? Cells ribosome, cells nucleus, okay? All of them can be used by virus. This is called cells machinery, can be used now by viruses. When it's ready to leave, instead of destroying the cell, the new virus just bud off from the cell. They're just going to bud off. Budding off means they just going to take off some membrane of the cell. Okay, some membrane of the cell. This is the outer membrane. Okay, outer membrane of the cell will be taken off by the virus. This is called now binding. Okay, so this is the envelope. You can see this is the envelope protein. Now they have the envelope. Now these viruses have envelope. Okay, so what's envelope? While binding out, each virus grabs a bit of the cell's membrane. Okay, cell's outer membrane. This is what this structure is called, membrane or plasma membrane. This plasma membrane actually can be taken up, can be grabbed by the viruses, okay? And they're going to wrap it up to itself, okay? This stolen membrane becomes the envelope for the new virus or around the virus, okay? Do you get my point? Everything clear? Now this virus is called enveloped virus, okay? Well, any questions? Why it's important? Why these animal viruses, why budding is important? So this is the cell stays alive for a longer period of time, right? Because the cell is not destroyed here, it can keep making more viruses like factory running over time. Okay, viruses with envelopes are harder for the immune system to detect. Why envelope is important for the virus? Because viruses with envelopes, they are harder for the immune system to detect. Our immune systems will tackle with that problem. So they are not going to kill this virus directly. Okay, it's really hard for the immune system to detect the enveloped viruses since because they are wearing a piece of what? They are wearing a piece of your own cell's membrane. It's taking off your own cell's membrane to the outer source. That's why your immune cells will get some difficulties while finding out this virus. Okay, okay, good. Any questions? Do you understand the point, right? So ligand and receptor here, like a key and the lock. Okay, budding means coating itself with the cell's membrane. Envelope, this is stolen code. Okay, good. If you understood everything, let's go to the RNA viruses. So what are the RNA viruses? So think of the RNA viruses like special types of the hackers. You know what's hackers? Hackers? Most living things, for example, us, plants, bacteria, they store their genetic code as what? As DNA. But RNA viruses, they are different. They carry their instructions in the molecule, which is called what? Which is called RNA. Okay, it's not the DNA. Some bacteria, some viruses, not all of them. We have the DNA, but viruses as well. We have the RNA viruses as well. Okay, so some RNA viruses, they differ from bacteria, plants, and us by containing the RNA in their genome. I said genome. Okay, humans, animals, bacteria, they have the DNA. Okay, which enzyme used for the replication in this case? DNA polymerase, right? We use DNA polymerase to synthesize new DNA from the old DNA. Okay, so what about RNA viruses? They use what kind of enzyme they're going to use to make more RNA? They're going to use RNA polymerase, not DNA polymerase. Okay, this is the special. Okay, why can't these RNA viruses use normal enzymes? In your body, you have the DNA, right? And this DNA in your body, imagine you have the DNA, and this DNA is copied, right? This DNA is copied using an enzyme. What? Which enzyme?
DNA.
DNA polymerase, right? By DNA polymerase. By DNA polymerase. But imagine. What about, but this enzyme doesn't work on the RNA. Imagine if I have the RNA, this enzyme is not working in the RNA, and that's all RNA viruses have. So these viruses bring their own special enzyme with them. This is called, they always bring themselves a special enzyme, and this enzyme is called what? RNA
RNA.
dependent RNA polymerase. Okay, RNA dependent RNA polymerase. It sounds very fancy, but it's what it means. So RNA dependent means it uses RNA as a starting material. We call it as a template. Okay, so RNA dependent means they're going to use the RNA molecule as a start, as a template. Okay, so RNA polymerase means it builds the new RNA strands. So DNA polymerase means it's building new strand of DNA. RNA polymerase means it's building the new strand of RNA. Polymerase means building. Okay, so RNA polymerase building the new RNA. RNA dependent means using the template of RNA here, not the DNA. Okay, good. Any question?
No.
Imagine everyone is using a charger. Everyone is using a charger that only works with Apple devices. Okay? For example, imagine the Apple is a DNA and the charger is DNA polymerase. Okay, everyone is using DNA polymerase for their Apple devices for the DNA. But RNA viruses, they're like Android phones. Okay, they bring their own charger. Imagine all your classmates use what iPhone, but you're using Android. What you have to do, you have to bring your own charger with yourself. Your own charger. Your own charger will be RNA dependent RNA polymerase. Okay, because the usual one won't work. I can explain in real life like this. Okay, so how it's going to replicate? Replicate means what? How it's going to increase in number. So the virus, first of all, enters to the host cell. It's going to enter to the cell. Inside, it uses its RNA-dependent RNA polymerase. They are making more copies. They are making more copies of these RNA molecules. And these RNA copies are used for what? For the protein synthesis. For the protein synthesis, serve as a genetic material for the new virus babies. We call them virions, okay? These viruses are built and leave the cell to infect the others. It's like the same with other cells, but the difference is only in the genome, only that they contain the RNA. Okay? So why it's important? This kind of RNA viruses, so RNA content, this makes the RNA viruses tricky because their enzyme is error prone. It makes a lot of mistakes. These mistakes are mutations which help the viruses to evolve very fast. That's why, do you know the flu virus? Flu degani prostuda. This is because of the virus. Shamollab qoldim deydi-ku, gripp bo'ldim deydi, shamollab qoldim deydi. Gripp degani bu bakterialniy infeksiya. Shamollab qoldim bu virusli infeksiya. O'shaning uchun that's why flu viruses change every year. Shuning uchun ham shamollashga har yili nima qilasizlar? Almashib turadi. Flu flu degani nima edi? Flu degani common cold degani gripp edi, flu degani shamollash edi adashmasam. Yeah, let me fact check. Just a moment. Flu grippe ekan. Mm. I'm very sorry. So flu, I'm sorry, confused between the cold, common cold, and the flu. Common cold это простуда. Let's write it down. So common cold, это простуда, and the flu is grippe. So when you get the flu, this is most, it can be bacterial, it can be viral. When you get the flu, so flu virus change every year. Do you remember that? That's why they every year they vaccinate for the grippe. Every year they're going to vaccinate, they're going to change. That's why it's hard to make the long lasting vaccines for some RNA viruses. And also, do you know the HIV, human immunodeficiency virus, which cause spit, we call them AIDS. It's also RNA virus. It's also changing. It's also undergoing mutations. So RNA viruses, they are tricky because their enzyme is error prone. It makes mistakes. Okay, these mistakes we call them mutations. Those viruses are always going into the mutations. That's why if you develop one vaccine in 2024, it's not going to work in 2025 because they are changing, they are mutating every year. Okay. SARS-CoV-2. Do you remember what SARS-CoV-2? Coronavirus. So coronavirus vaccine, every year they are also doing updates every year. So that means that this virus is also changing. Do you read the news saying that the coronavirus, new mutations, new strands are coming, they're developing again, new pandemic is going to appear. So that's because the virus is going to change its information again and again because it is enzyme. RNA-dependent RNA polymerase is enzyme. They're error prone, so they're going to make errors while making the new RNA strands. Okay, so HIV, SARS-CoV-2, they're all of them RNA viruses. Coronavirus, HIV, and flu, which we call grippe, all of them are viruses. Okay. So DNA polymerase works only with DNA. RNA polymerase, they need RNA viruses. They need RNA viruses to work. Okay. So RNA-dependent RNA polymerase makes the new RNA from the RNA molecule. RNA virus means a virus, those genetic material is RNA. Okay. Examples to the RNA viruses, influenza virus, which causes what? Which influenza virus, which causes the flu, coronavirus, also measles. What's measles, you know? Qizamiq deb eshitganmisizlar? Qizamiq. На русском будет ветрянками, оспамidi hozirsan. Let me. Да, корь, точно. Корь. Корь, свинка красного. Okay. Corey. This is also what? RNA virus. What about Ebola? Have you heard about Ebola in the Africa? It is also RNA virus. So RNA virus is dangerous. Okay. Good. Any questions regarding the RNA viruses?
You said that RNA viruses changes, right?
Yes, changes every.
DNA viruses also change.
They don't change as much as RNA viruses. RNA they change almost every year. There is no cure for the RNA viruses still. They're always changing their information. Okay. But for the DNA ones, we have, for example, hepatitis A, we have a cure. Hepatitis B, C. For the D strand, for the B strand, we still don't have. So we're doing the research. But if we launch it, if we do it, so we can get good results and it won't be changed for a long time. For sure. Questions? No, if no questions, we can shift to the retroviruses. So retroviruses, they are also RNA viruses, but instead of copying the RNA like normal RNA viruses, right? The RNA virus copied its RNA from the RNA, right? Instead of copying these retroviruses, instead of copying the RNA like the RNA viruses, they do something backup. Backup means what? Retro. Okay, they're going to do some backup, which means retro. So retro means backwards. They turn their RNA into the DNA. Okay, so they have the RNA. They're not going to make the RNA from their RNA, but they're going to make DNA from their RNA. Okay, so why retro? Because in nature, there is a central dogma which goes from DNA, we always synthesize RNA, right? From the RNA, we always synthesize what? Proteins, right? We always synthesize the proteins. Why retro? But retroviruses do vice versa. From the RNA, they're going to synthesize what? DNA. From that DNA, again, they're going to synthesize the RNA, and then they're going to synthesize the protein. Okay? They reverse the usual direction, and that's why they are called what? Retroviruses. That's why we call them retroviruses. Good? Um, okay, understandable, right? And then we learned why they called the retro. So how they're going to work, let's go step by step. The picture, regarding the picture, you see the retrovirus enters to the host cell. So this is the retrovirus. You see, this is the retrovirus. It's entering to the host cell. Okay, well, what it's called? It's called endocytosis. Endocytosis moving, it's in the bulk transport, we learn it together. Moving of the larger substances into the cell is called what? Endocytosis. Am I right? Endocytosis, exocytosis, this is going outside of the cell. There are two types of the endocytosis, phagocytosis and pinocytosis. This is phagocytosis mostly, okay? So it releasing its RNA. Now, this capsule is going to break down inside of the cell, and this is what? This is the RNA inside of the virus, okay? This is the RNA molecule, okay? It brings along the special enzyme, which is called reverse transcriptase, okay? It's doing what? It's bringing with itself, you see, it's bringing, this is the reverse transcriptase is inside, it's bringing with itself a special enzyme which is called the reverse transcriptase, okay? This enzyme turns the RNA molecule into the DNA. You see how they respond? This enzyme reverse transcriptase is going to turn the RNA molecule into the DNA molecule. The new DNA is now called the proviral DNA, and then it's inserted into the host's DNA. Do you see? This DNA now is inserted to the host DNA. This is the host's DNA, and it's inserting to the middle of the host DNA now. Can you see it? Now it's in which enzyme is helping this DNA molecule to enter to the nucleus? We have a special enzyme which is called integrase, which helps this DNA molecule to enter to the host's DNA. Now the virus becomes part of your cell's DNA. Now it's the part of your cell's DNA, okay? Your cells unknowingly, your cells are not knowing, they unknowingly reads, that means transcribing the viral genes and making more RNA copies and more viral proteins, okay? So transcription is happening. Transcription is meaning from the DNA synthesizing the RNAs, right? We are copying the DNA, we're getting this information of the DNA and making the RNA molecules. We call it messenger RNAs. So we are making more messenger RNAs. That means we are making more viral RNA, more viral protein. The new viruses then are going to assemble, they're going to assemble and bud off the cell. They're going to tuplan out. They reform as a virus, okay? They're going to synthesize protein, which make the capsid, and their new viral RNA is going to enter to the virus, okay? They're going to enter to the virus. Now you see, in order to multiply, in order to become more, they are using your cells. They integrated their information to the DNA to make more RNA. Every time when the protein synthesis happens, when DNA transcribes, it's going to synthesize new virus. Your cells, machinery are used by the viruses to make more viruses. Okay, the new viruses assembles and they're going to bud off. They're budding off. This is the new virion, and they have what? They have the envelope, which is really hard for our body to detect them. It's very hard for our immune cells to detect them because on the surface we have our own what information, our own cell information. It's because it came from the envelope, came from our own membrane. Okay?
Mhm.
It's like a virus is spying here. Virus is entering to your home, which means your cell, writing its own instructions like to your diary. You have a diary, which is your DNA. It's writing, virus is writing, the spy is writing its own instructions to your diary. Now, every time you follow your routine, you're also following the secret plan, which was written by your spy, which is virus. Your cell is doing what the virus is telling every time, okay? Example to this, I can say also HIV. That's why there is no cure for the HIV. HIV, the virus that causes the AIDS, spit, okay? The most famous retrovirus is HIV. Okay, that's why HIV stays in the body for a long time, okay? Until your lifetimes, HIV is going to stay in your body. Its genes are built in your cell's DNA. Genes of this virus is already in your DNA. You cannot remove it. It's already integrated to your DNA. This also makes hard to cure. So that's why it's very hard to cure the HIV, because the virus is hidden inside your own genetic material. Okay? Any questions? So retro means reverse. Retroviruses reverse the central dogma of the biology using the reverse transcriptase. That was all about our today's lecture regarding the animal viruses, retroviruses, and animal RNA viruses.